So I'm going to talk about Snudda, which is a software we created to generate microcircuits, which can be run in Neuron. And the key thing here is that we want to use the morphologies, the axons and dendrites located in space to determine where we should place the synapses. And this is collaborative work within the lab of Jeanette Hellgren-Kotaleski. We worked closely with Sten Grillner, and we have experimental collaborations with Gilad Silverberg and also with Kai Du in China. And we have 3 publications that if you want to have a closer look, you can find them here. So let's start with, with the pipeline. So the pipeline consists of different parts. Most of these morphologies that we use come from either experiments that done at KI or from neuromorpho.org, and they might not be complete. So we have the first step is morphological repair using tree, which can sort of take a morphology which has been flattened and expanding it and looking for cut neurites and try to sort of extend them somewhat. Then we use BluePyOpt to do the model optimization. So to generate a population of models that we can use. And then Snudda comes in, which creates the network by doing touch detection. And finally, we run the simulation using Neuron. And this can be run either on laptops or on larger computers. So yeah, so Snudda does voxel-based touch detection. The word Snudda means to touch, lightly touch in Swedish. We have a rule-based pruning algorithm to reduce the putative sets of synapses that were detected by touch detection to actual set of synapses that are used in simulation. We also have some scripts for network analysis and then finally to run the simulation. And all of this is open source and you can find it on GitHub. You can also do pip install Snudda to get it. So to run this, you will need morphologies, which dendrites and preferably also axons. So as complete morphologies as possible. You will need electrophysiological neuron, which in our case are optimized by BluePyOpt. So we need recorded data to do this optimization. You will need synapse models because, of course, these neurons are connected by synapses. You will have detailed synapse models with facilitation, depression, things like that. And finally, you need connection statistics. So basically, quite often pairwise recordings of multiple pairs. And then you can know that maybe 1/3 of these neurons within a certain distance are connected to each other. So the touch detection is done using a voxel-based method. So we voxelize the space into voxels which are 3 by 3 micrometers large. And then we basically see each axon, each dendrite for every neuron, what voxel do they occupy. And if there are an axon and a dendrite from different neurons occupying the same voxel, there's a putative synapse there if the neuron types allow that connection. So here you can see just schematically what it would look like. And then in our voxel representation. And then finally, when you're done touch detection, we can do Blender renderings to show where the synapses were located. So the structure we're looking at is the stratum. So that's our sort of the case of interest. And the entire mouse stratum contains 1.72 million neurons. And you can do touch detection, you will find about 31 billion synapses. Oh, sorry, 31 billion putative synapse locations. So here you see the stratum. Here's a voxel or a hypervoxel. So it's basically a cube containing 100 by 100 by 100, so 1 million voxels that we use for touch detection. And it's about 4,000 neurons usually within that volume. And if you would render all the axons, so this is just the somas here, and if you would render all axons and dendrites, you can see this quite closely packed volume. So the basal ganglia and the stratum is what we're interested in, and there's 2 different neuron types. Sorry, I should say there's multiple neuron types, but the main neuron types are the spiny pressure neurons, which are 2 different types, the ISBN and the ESBN, and they make up approximately 95% of all neurons. And the remaining neuron types are interneurons, and in particular fastback interneurons, CHIN and LTS are the ones we include in our models. So as I mentioned before, you need to have connectivity statistics, so pairwise recordings. And in this schematic figure here, we show that, we show the connectivity between, for instance, the DSP and the ISBN. And the data you can see here in between shows what is the connection probability measured in experiments. And there are 2 different experiments, one which was within 50 micrometers those are in red. Another one we had measured within 100 micrometers, those are values in blue. So we have those values, and what we want to do then is we do the touch detection, and then we want to tune our pruning parameters to match experimental data. Because once you do touch detection, you get a lot of putative synapses. So here we have experimental data showing, for instance, DSPN to DSPN. The red lines correspond to the experimental data that's been measured. And the gray line is after the pruning step, how many connections do we have left? What's the connection probability? And you can see the gray lines here. So this is DSPN to DSPN, ISPN to DSPN, FS to DSPN, LTS to DSPN, and CHIN to DSPN. So this is just a subset of all the possible combination of neurons that can be connected, but just to show you. And we also, in the pruning rules, try to make sure that we have a number of synapses connecting pairs together that are match what we see in reality. So you can see the corresponding statistics here. And then because, sorry, and then because what we have is we have realistic densities of neurons. We can also, based on the connectivity, etc., we can derive what is the number of DSPNs that connect to other DSPNs within the neighborhood. And then finally, of course, one step to sort of see, because once you run a simulation, you want to make sure that when you stimulate a DSPN, that you have a corresponding appropriate stimulation in nearby neurons, that the synapses are not too strong, that there are not too many synapses that become too strong, the connectivity. So we do virtual experiments where we inject current into a neuron, and then we record from the neighboring neurons in the network, and we see what is the depolarization or hyperpolarization that you see. And then, excuse me. And then we can run this in large-scale simulations. So here you can see a network simulation with straight line neurons, DSPN, ISPN, CHIN, LTS, and FS. And in this case, we had the base activity of cortical and thalamic. And then we had a cortical extra stimulation, a cortical command. And here at a later stage, we have dopamine activation. And the interesting thing here is that the DSPNs will become more active and the ISPNs will become less active when you have dopamine added to the system. Okay, so if you're more interested in finding out about the Snudda, we have a set of Jupyter notebooks, which goes through different use cases, how to sort of create simple networks, how to basically place neurons, to touch detection, pruning, and then eventually simulation of the network. So if you go to GitHub here, you can find out more about that. Oops, you should click here. So if you then go here, you can see that we have simple examples to create networks. We can create slices corresponding to the in vitro slices. You can tag the populations. You can have different population units. You can define networks where you have varying densities throughout the volume. You can have sort of preferred orientations of the neurons within the volume. You can also sort of have different volumes you created together, created and then connect them together. Examples for how to set up input generation for the simulation later on. Yeah, and then examples for stratum and also visualizations of this. If you are curious, a little bit more curious, I have a video presentation from the INCF conference. So there's a QR code here, which you can just go to, or you can go to GitHub and you can also find a link for the videos. Yeah, so there's two ways to run. One is to using a Jupyter notebook, and the other way is to use command line. So this is just a very, very quick example to show you that you can start, you set up some environment variables to run IP parallel, and then you start IP cluster. You define the network here, and then you can say, I want to place the cells, and then it places them. And you run detect, you run prune, and then you need to copy over an input JSON file. This is a specification for the input files. And then finally, you run the inputs, and sorry, and now finally, you run the simulations. So let me just jump back to this screen. Yeah, so here's some information basically for what you need to do if you want to install it. It's basically either it's pip install Snudda, or you can go to the repository and clone it from there.